A very good morning, class. We have 38 participants. I hope the rest will still be coming in soon. But anyhow, um, we will start class as usual. So for today's class, we are going to be learning about the different kinds of literature search and how we can use museum collections as a part of your reference for um, in regards to understanding the collections that you have. First and foremost, I would like to know whether you all can hear me well and clear.
Yes, Doctor.
Clear, Doctor.
Okay, all right, that's good. Thank you very much. Yeah. Okay. So first things first. Just a little bit of the recap of what we've learned. We have learned about when you've collected samples, what you have to do when you collect samples. You have to sort, you have to look at its morphological features, you have to identify them. Then you have to verify them, say on a platform like Worms, to see if the species is the one you're talking about, is the name correct. And then you have to make the decision of not whether or not it's a new species or this is a species that is found in our waters. So this is sort of like the step-by-step process. Now, with regards to some of the mistakes or bad examples that are sometimes practiced in the world of taxonomy, okay, taxonomists discourage description from non-taxonomists and non-specialists. Okay, we discourage, we don't hate them. um, Especially when it comes to ecologists. Again, at least in some ways, ecologists probably have, to some extent, a better, closer understanding in comparison to, say, those in the biotechnology field or anywhere where it's really out of the discipline. Okay, why we discourage that is sometimes everything is pooled into one species example. Okay, so this can be a problem because, as most of you might already understand from now, every species has its own differences in terms of its biology, in terms of its life cycle, in terms of the habitat it stays in. But that is why we had to understand that speciation lecture in the one that we learned on Sunday, because you know that there's certain gray areas and there's considered to be ever evolving. So because of that, sometimes if you were to pull everything together, and if a particular species is something that has some direct impact to us, for example, then for example, if you're retrieving a certain medicinal properties from the species, then it can be a problem because if you're not dealing with the right species, it can be actually toxic to us rather than beneficial. Okay, that's what we're putting out there. Or in the case that we assume that this species can help be somewhat like a new keystone species for, or to help reduce certain pests in a particular environment. However, if we introduce, say, the wrong species into the environment, can become invasive rather than beneficial for the environment. So things like that, sometimes we discourage description because as a taxonomist, we look into the identity of the species from both morphological or molecular aspects. Sometimes even those on the molecular aspect would just want to look at like the fingerprint without having to understand the identity. So this is quite tricky, which we cannot encourage such things to happen. As much as possible, taxonomists, we like to have full-on face value of every aspect that we're talking about of our subject matter. So mistakes are easy to make when using incomplete material. Okay, again, when you're looking at a particular species of interest, you want to be as thorough as possible in terms of its identity. So sometimes you can, in terms of incomplete material as fossils, of course, fossils are beneficial, but at the same time, there's so many aspects that we don't know, and we have to assume based on the available evidence that there is. And sometimes you have to make comparisons of fossils with current living organisms, which is probably closely related to it. Okay, but this is always difficult to explain sometimes, because simply put, when you have a fossil of a dinosaur, if you're trying to sort of remake how the dinosaur might look like, what we assume in terms of the colors might be actually different from its original aspect. So certain things can be a little bit incomplete. Okay, smashed and fragmented specimens from dredge or trawl holes. This, of course, it's very much difficult sometimes, and especially for students who are not familiar, you might assume that this is something new, but it's actually, say, part of a fragment of a fossil. Of a, say, coral, or it's just part of a limb of an animal, or it's just part of a segment of a polychaete, or it's actually just might be just eggs of a shark, but you might assume of it to be something. There's so many things out there. It can be, and when it's smashed and it's fragmented, it becomes even more difficult to us to identify what it is. So smashed and fragmented specimens are usually not very helpful in these matters. Then not recognizing that some item doesn't belong in a sample at all. Very simply put, sometimes I think a lot of you may have experienced when you're looking through a microscope, you might actually see some very interesting stuff, in your specimens, but not realizing that that is actually a form of microplastic. Okay, but when you first see it, you may not know. So these are things that are bound to happen if you're not experienced in that matter. Okay, and of course so ultimately it all boils down to having a poor understanding of the species itself and its ecology or its biology. Yes, taxonomy can be very tricky in a sense that taxonomy study years. It's almost like a person who's studying cancer. You study years of trying to resolve the issues with cancer. And then you have keyboard warriors who are trying to explain what this is or what medication can help. So that is how sometimes taxonomists do feel. But we have keyboard warriors online trying to resolve, this species is so and so. Like the case of there were some issues where they found, say, example, like a nematode in a tin. Okay, but maybe it looked like some form of other crustacean. So you have all sorts of species of crustaceans, but these are not experts actually trying to explain about it. So these are the things that sometimes we make bad assumptions. A perfect example is like on Facebook, when people try to give feedback, but you don't have the right expert explaining the problem. Okay, so which is why it is very, very important to establish an identity for when it comes to taxonomy. Okay, so one of the things that we look at is, of course, as much as possible, if we can go into genetics, it is good to have that profile for genetics, because from genetics we can see whether certain aspects of the specimen of interest, is it a variant or is it mutant? Okay, and then variant produced by under unfavorable conditions. Okay, so remember like how we explained about that speciation example of uncontaminated soil and contaminated soil, and how some plants actually were able to adapt to this new environment. So again, was it more of a variant that was produced in those kind of unfavorable conditions? So other examples include plant which is subjected to heavy grazing, or animals which are stunted by inadequate food supply. So some may thrive, or some might actually not thrive. And then when you're looking into establishing identity, you want the most recent description, preferably not in a book or article used for identification. What they mean by this is that sometimes picture guides or field books for to the novice, or to a novice or to an amateur, for first timer, it's okay. But if you want very detailed descriptions about this, a book and an article is not the best means for understanding description. Preferably, you need to go to a proper literature. Like in our case, it's sometimes we use journals like Zootaxa, Zookeys, Biotaxa. There's so many other taxonomic journals available out there. which is most helpful for your understanding. And then it's also important when establishing the identity, known species elsewhere but not worldwide. Okay, this is what one term as new record, meaning to say that the species was already found by somewhere else. Say it was discovered in Japan, but then later it is found in, say, example, Malaysia. So it's not entirely a new species, but nobody else reported it in Malaysia, so it's a first time record. So it's a new record. But also we need to understand that new record, did it by chance, is it occur naturally there? Or is this species introduced? So those are things you want to also understand, because if it's introduced, it's different from it being endemic there. And then also we want to know if it's rare or inconspicuous and overlooked by earlier workers. Okay. And then of course, if you establish all the different aspects you have to look at in a person's identity, and you realize that it's not a new record, then of course you definitely found a new species. And your work is to name and describe this new species to the best possible detail. But then here we have a problem, because there are times that a species can have been described and reduced to its synonym. So the later worker decided it's a species variant from an earlier name and placed it in a list of alternative names. Or the original name is no longer used and the original name is reinstated. Okay, but there's also another condition where it was previously known but called the wrong name. Okay, so a redescription and a new name was indicated. So example, early naturalists gave many New World species names belonging to species distributed in Europe and Great Britain, but later more detailed study found that they were actually very distinct. So you have conditions where people thought that certain species are the same, but they're actually very different. And then you also have conditions where species are different, but they were thought to be same. This is what the summary of this work is about. Okay. So what exactly do you have to do when you're faced in this kind of situation? So, of course, the first thing first is to track down descriptions of closely related species in literature. So just like any normal scientific study, you have to start off with a literature review or literature search. So likewise in taxonomy, you have to track down all possible literature, from guidebooks, field books, right up to the most detailed journal descriptions of it. And then learn to read or ask an expert about it. So while you're reading, you will obviously notice when it comes to journals, certain names are very commonly written. Those are authors in the field of expertise. So that is in reference to an expert. So an expert is really looking into basically their Google Scholar and their Scopus, and then seeing what other sort of literature work have they done. How active are they in that field of study? So those are experts in the field that you really want to ask their opinion. Okay, so like in my case, when it comes to overall crustaceans, one of the best known people like globally is Peter Ng from Singapore. Okay, so the moment the name Peter Ng is mentioned, people go like, oh wow, we kowtow to him, you know. So that is where you want to establish your kind of work. And of course, when reading and talking to an expert and talking to a curator like Azwarina herself, then of course you want to locate and examine the actual specimens. So these are things I apologize for the third year students. Maybe this was not explained to you very well in detail, because a lot of what you did for your kerja lapangan is, or your field work, is that you collected your samples and you identified and made some form of reference to, say, Wild Singapore, etc., to our neighboring, to our neighbors that actually have some form of reference. But the actual process of ensuring what we identify is true is actually to look into the type specimens, which Azwarina has mentioned before. And this means that you actually have to work in a repository or museum to see, is this correct or not? All right. So when you're looking in terms of the taxonomic literature, the taxonomic research itself, where are you to find this sort of taxonomic literature? All right, so one of the first places you can find is university library. Depending on how established your university library is, if you have like, like Smithsonian Museum, which has their own library, but or university library that actually works very well with the kind of specimens you're working on, they would actually have a complete collections. I do apologize in the sense that UMT has a little bit of it, but not in the Malaysian context, because in Malaysia, we still don't have very many taxonomists working in their respective fields. Okay, so our collections is still very minimal. So this is where we've identified some gaps in this matter. So this is something that we need to establish more, particularly because UMT is actually nearby marine waters. So by right, their cataloging and also their collections for marine or aquatic related research should be more established. Okay, then of course you can visit museum and research institutes. So there are some that is actually available online, and then of course if going to the museum itself, you will actually get to look at their whole collections. If at any chance you guys get to have an internship, say in Australia or the Smithsonian Museum or in Paris, the museums are such is it's so big. It's not only do you see an exhibition in the front, but you have a level which is just solely dedicated to your research materials. So it's very interesting to see such available literature there. And then, of course, for taxonomy, one of our heavily used online sites is the Biodiversity Heritage Library. I will show you that very shortly. And also online taxonomic journals. So like the ones depending on whether you're doing on zoology or botany or on bacteria or virology, it's actually quite different. Okay, so as you would understand by now, or you've at least experienced, that there are different types of taxonomic literature. So you have general guides. A general guide is a quick guide to the biological literature. And then you have books and textbooks. So they are only looking more detail into the biology, but there is, or looking into generalization of higher taxa. And then you have field guides. Field guides help with field sampling. And then some are actually helpful that they can come with keys. And then you have abstracts and indexes, like it has a record and biological abstract. And of course, when you're talking about taxonomic descriptions that I mentioned before, you want to look at the taxonomic literature. Okay, so I don't know if you can see my video. I've turned on my video. Are you able to see me?
Yes, Doctor.
Yes, sir.
Okay, so I want to give you some examples. Okay, so when you're looking at a book and a textbook, okay, for undergraduates, like you have no idea, like say what selves is or what, certain marine fishes are. So this is what you mean by a textbook. So a textbook gives you a basic understanding of the biology of the work. Okay, so I'm sure all of you are quite familiar with this book, right? So your textbook talks about, things like this. It gives you a lot of pictures, but also a bit of understanding of the species that you're talking about. Okay, so it gives all its general anatomical structures, that's all. But in your taxonomic journal, basically you won't have all of these detailed descriptions because technically if you're working as an expert, you already know those anatomical structures. But say if you're very much interested in, say, working on copepods, so obviously you want to know how to differentiate a female from a male, etc. Okay, so this is an example of a textbook. If you're talking about field guides, okay, so this is an example of a field guide. Please let me know if you can't see, yeah? If I'm showing this, I'm assuming that you can see. So this field guide, of course, is released by Jabatan Perikanan Malaysia, the Fisheries Department. So these field guides are important because on the field, obviously you don't want to be doing intense reading. You just want to be able to look at the specimen and quickly see if you can make a quick search. All right. So your field guides will actually have a quick quick idea of what the species is about in terms of how you can identify it. So it talks about, okay, this species, you have to look at this caudal fin, you have to look at how many vertebrae there is, etc. Something like that. Okay, so this is what field guides are supposed to be about. It doesn't have a detailed biology or life cycle about the work like that of a textbook. Okay, so when you're starting off in terms of reading something, those are things you want to start off with if you have no idea about anything about the species. Okay, another form of field guide is like this. Okay, this can be quite helpful, I believe. Yeah, okay, when I talk about using a key, so I think most of you have seen this. So this is quite helpful because there are certain keys that actually uses an epithet of morphological statements. It's meaning to say that they say fish with fins and fish without fins. If fish with fins, you move on to 2. If fish without fins, you move on to 4. Example. But this one is quite interesting because their keys is looking into morphological features. So if morphological features, if you have something of this shape, it'll be of this family. And then they'll tell you what pages to go to or what plates. So this is what field guides are supposed to be like for you. If you're looking at I didn't mention here about checklist, okay, but it's almost quite similar to abstracts and indices. If it's a checklist, usually have a short introduction about the family that you're working with, what sort of organisms, and it's more catered to a specific location. Say, example, a checklist of moths from Tasekene. Okay, then what it will give you is sometimes it might even give you statistics about what they're likely to find, and this part is the most important. Okay, so a checklist will actually give you the family, the species, and what they found. Some styles of checklists is also a bit different, where, like, say, this one on the diversity and abundance of fish assemblages in situ wetlands. So generally, give you a short introduction, and then some might even give you a map. But and there's some even include how you use their ecological indices. Like in this case, they're using biodiversity parameters using primer, and then this is their checklist. So their checklist will include things like family, species, local name, English name, and this is interesting. They even mention about IUCN status and percentage abundance. Okay, so this checklist is it's a little bit more advanced, like in a way that you are familiar with some of the species, but now you want to know in the context of, say, Malaysian waters. So this will be like a quick guide to, okay, this is what species are available. But again, depending, so this is a more, imagine you're starting off all of this at the beginner's level. This is more on a immediate level because you want a quick look of what species you're dealing with. But if you want to go to the advanced level of example saying, okay, like these people found Sardinella gibbosa and Sardinella fimbriata, but how likely are those species to be named correctly as so? So you want to be able to look at the specimen. Now the thing with certain checklists is that some of them do not have the registration details of the specimen. Okay, it's just a catch and a release thing. So preferably if you have a checklist that actually has the registration and where it's deposited in. So example, like if I were to write, okay, all of the specimens were found in RRC. So at least you yourself can actually check at RRC and check with this registration details, like the SRN that you guys did, so that you can see, hey, is this true or not? What you've collected in the field and what is available in the museum or the repository. Okay, so pretty much, okay, it's pretty much abstracts also are like that. And there's another thing, I kind of forgot which one is it, but probably I'll come towards the end. But this is what it is about. Taxonomic journals, I will continue in a later lecture of what to do so that we can break it down, because this one will actually take some time. At this stage, are there any questions that you want to clarify with me? Okay, very good. So you're clear, yeah? Okay. So I will cover how to read journals in the next lecture, but a typical structure of a lecture would be a journal, sorry, would be such. You will have a heading of the family of interest, okay, and then we'll have the genus, and this is the author and the year that we have. So then you will have the name and the figure. So this is the name, the species name, and then you will indicate whether it's a new species or not. And where are those figures found? Figures in a sense whether you've taken a photographic picture or whether you've done a digital drawing or a hand drawing. What are the labels of the figures? And it will be placed later in the text or probably after. So your diagnosis, just like how you have a medical diagnosis, you also have a diagnosis, meaning how do you quickly identify the species. So example, if you have like the Sardinella, you have 10 different species of Sardinella. The description part will give detailed explanation of, say, Sardinella species 1. You know, it talks about the eyes, the fins, the caudal fin, etc. All detail for all of the 10 species. But diagnosis will actually pick out what is the most important feature that you can quickly identify species A in comparison to species B, C, etc. That is what a diagnosis is. Okay, the type, like Ms. Azwarina has mentioned before, is the reference of that specimen, and it's not just any form of reference, it's the ones that the authors described it of, and that is deposited in the National Museum in the US with this naturalization code. Etymology means how did the authors name this species? Okay, so they mentioned here that is name for the country of, I can't quite see here, Belize. Okay, so it's found in Belize, in which the species was found. So that is what etymology means. How did the species name came to be formed? And then you have a discussion. Discussion usually talks about how is a species different from other species. So it will actually explain about, it will reference other species in preference, and if there's anything unique about this in terms of if the species was discovered before, are there any particular issues that you want to raise in terms of the naming system, like where was it formerly named under and why it had to be pooled and all those kind of things. Occurrence is the geographical occurrence or the distribution. Some might even give on colors and sizes as well. Okay, so if you want to find a literature, remember I mentioned that you can find online literature. So one of our most used platforms is the Biodiversity Heritage Library. Okay, so you can find latest scientific journals, but you can also find some very old papers, right up from 1780s, example. So for me, when you're looking at this platform, you can actually browse by either the title, the author. The date is not going to be very useful, because if you just have a date, you might actually have a long list of so many other papers on that date. So it's best to go with the title or the author. All right. Collections and contributors, if you have those details, contributor is who have actually uploaded the work, which library has done so. If you can identify, it's great, but otherwise, for beginners, I will go with the title and the author. So for example, one person that I heavily use when it comes to working on my specimens is Shodet and Minute. It's a collaborative work. So it's Frederick, his name is Frederick Minute. So when I type Frederick Minute here, so if you're going to use a general search button here, it will give you all sorts of publications, but you're going to have to look through a whole lot. And then you have it also search of the author that you're talking about. So this is an easier search, because if I click on this, it will lead me to all of the publications by this particular author. So they've actually uploaded some of all of their scientific work. So in my case, I'm looking, okay, Malacostraca is not what I'm quite looking for. So I'm looking, say, example with the isopods. So if I look on isopods, if I click on that, basically it'll give me a whole list of the, all the pages within this book. So the only thing is, yes, so usually the style of how these taxonomists used to do their work is all of the pages are the species of what they're talking about, but towards the end, they will actually put in all the illustrations. And if you notice here on your left, they use the term tab, or they would use the word plate. So these were the drawings at the time. So it might look like, meh, it's not that interesting, but you have to appreciate work back in the 17 and 1800s, okay, when microscope was just being established or just innovated at the time. So this is the best detail they could draw. So it's like walking through time, through history, and it's really appreciating that kind of artwork. It's really nice, but of course, now with modern taxonomic work, it's a very different approach. But considerably at that time, it was one of their best works. And this is the contributor, Smithsonian Libraries. Okay, so we really ought to thank a lot of these libraries who've painstakingly uploaded this work so that globally we can assess to see whether the species we're talking about is actually correct or not. Because otherwise, you'll be blindly naming all of the species without really being sure whether all of these authors have actually found this species before or not. So mind you, somebody may have found this back in the 1700s, and you might think that you actually found it as something new. So you can't just simply take credit for your own new work and not give credit for people who have actually published this work, even though it's coming from the 1700s or 1800s. As long as it's published, you actually have to give credit for this work. And of course, things were written in Latin. So yes, if you can actually read Latin, it's good. But now it's such that you can actually highlight some of these words and then you can do a Google Translate. But if you're actually familiar with some of the words, it can be actually quite useful. Like, of course, this is length. So it's talking about how many millimeters it is in. And yeah, actually it's quite, I must say that I'm not actually very good in Latin, but certain things I can understand, like what carina is and the coda, peralta, immersa. I'm quite familiar as you read up some of this. So yeah, if it's femina or vigera, it's ovigerous female. So if you're talking about this species in specific, it talks about which tab you can find it in and which figure. So that is basically what you can do when you're actually looking at the biodiversity heritage library. I would encourage you, if you have time, to actually play around and look into what you're doing. Okay, so if you've noticed that taxonomy, I hope now you have a better understanding that it's not just about looking at a species or looking at a picture guide and going like, this is the species I'm talking about. Ta-da, it's taxonomy. No, it's a lot of CSI work, crime scene investigation work. You have to keep a diary and you have to make an autopsy. So again, your processes, a summary of what we've been talking about. So when it comes to sampling, so you've done all your sorting, your new starfish, your sponges, etc. Then you stumble upon this new polychaete. And then you go, do you think you found a new species? So that's where the whole literature search first comes. So remember all the different kinds of literature. If you are a beginner, you have no idea where to start, of course you have to start with your textbooks, you have to start with your field guides, move on to checklists, and then move on to journals. So for looking at all this, you realize, no, you have not found it at all. So there's nothing in all literature in the database. Then, of course, hooray, you found a new species. So all you have to do is go and register it. But say in the literature, and then you found, hey, okay, you found it. Now, the next step is actually looking at, is it similar to your type or non-type? Okay. Now, the thing with literature is sometimes, if the description is very complete, like with a journal, that you're able to immediately identify it, then it's fine. But if certain things you're feeling unsure, like it may be like a variant, or it may be a good enough point to make it into a new species or what. So this is where you have to look at the collections, okay, especially if there's available collection. So from your specimen, you have to compare, like say, example, going to RRC. Is it similar to your kind of specimen, or the type specimen or the non-type specimen? So if it's not the same, then hooray, it is technically also a new species. But if it is, but maybe it's not been looked into for a very long time, you can either do a review for the species, you can redescribe it, or you can complete its identity. Again, this is dealing with experts and all to help guide you through what to do.